See if you can from memory. Oops. Okay, see if you can from memory, draw a eukaryotic cell. Draw, label and annotate a eukaryotic cell. Okay, this is something that was on a couple of your tests last term. It is something that you do need to be able to do as part of the diploma. I do it in pencil or erasable pen if you can. Always, drawing's always in erasable pen or pencil. Okay, and let's see how close. I'm going to draw one in here as well. Let's see how close you guys get to mine. So a eukaryote, not a prokaryote.
No, you serious? No, there's a little tail I think prokaryotes. Yeah. The little.
I'm going to do this on the board actually because I hate drawing the pro lines. Okay, so the key here is leave yourself some space for your annotations. Okay, draw, label, and annotate. Let's move this. Old technology, always the best when it comes to these sort of things. Anyway. Okay, I'll make a start, but don't copy mine just yet. Pretty good, guys. Didn't think you'd get a test on your first day back, did you? Again, this is all feedback for you to tell yourself, thinking about what you can remember, what you know, what you might need to review. Excuse me. Okay, so when you guys finish, what I'd like you to do is take another colour, maybe like a red, something that's going to stick out, and correct what you've drawn if there's any mistakes compared to the diagram that I put on the board. Does that make sense? Mm-hmm. Okay, so just critically look at what you've done. Again, I haven't given you any chance to review this. This is just purely straight out of your memory. So if it's all right, that's really, really good. It's excellent. Okay, if there's a few mistakes, that's not a problem. Not a problem at all. But just use it as an opportunity to review it. So just take another pen, another colour that's going to stand out and review your work against what I put on here. Okay? You might be thinking, what has this got to do with proteins? I wonder if there's any Any bright spark on a Wednesday morning, first day of the term, who could tell me why I thought you should do this? Do all of them. That's they get the idea. Looking quizzical. What are you thinking about?
No, I'm just thinking about a name of one thing.
Okay. Am I missing anything?
I don't think so.
Yeah. One of those. Yeah, is that what you're after? Yeah. I could actually add something like that to here.
Yeah.
What are they called? Vesicles. Well done. Yeah. Good.
One thing called was it?
There was also the lyosome.
Yeah, good. I'm not going to include that just because I want to keep it. I have got a purpose for this, but you're right. There would be a lysosome as well. Yeah. And there's other things that we haven't included in the diploma course, like the cytoskeleton. Basically, we've peeled back one layer of the onion more than the MYP, but if you were to do this at university, there'd be even more structures that you would be introduced to. Okay, so we're finding a happy balance. All right. Okay, so let's see if we can add some labels to this. Is this an animal or a plant cell as a start?
Animal.
Yes, thank you. Excellent.
Mitochondria and membrane.
Yeah, but plants have those too. Why is it not a plant cell? What's missing?
Cell wall.
Cell wall would be good. Okay, and what's the carbohydrate that makes up cell walls? Begins with C, has an E in it. Sounds like cell. Cellulose. Yeah. Okay. So plants have a cell wall as well, made from cellulose, which is a carbohydrate. We've also got a large central vacuole as a chloroplast. I haven't included them just because I haven't got so much space. Okay. So this structure is plasma membrane. Good. Plasma membrane. Okay. I'll add the annotations in a minute. Okay. This area that's kind of white would be the cytoplasm. Good. Okay, great. This structure here, the HQ of the cell. Nucleus. Yeah, okay. Notice, though, I've drawn a double membrane with little holes in it, pores. Okay, it's a two-dimensional representation of something that would be 3D. Okay, so this is our nucleus. Okay, and this is a nuclear pore.
Oh, do you have to include that?
I would include that as well. Okay. The most important thing is that you show it with a double membrane. So if you do this in an exam, the IB will expect you to show it as a double membrane with pores, and the pores are really important, and you'll understand why they're important when we get onto genetics. Okay. They are really important. When we get onto the sort of genetic and molecular, when we look at DNA, you'll begin to find out why those are really important. Okay, but for now we'll just leave it as that. Okay, these dudes, these kind of dots.
ATs, ribosome.
ATS ribosome, so free ATS ribosomes. Good, well done.
Free.
I'll explain the distinction in a second. Okay.
Shall I write it down?
Yeah, we call them free ATS ribosomes. They're free as in they're not attached to anything. Whereas these dudes. What are they attached to?
The rough yolk.
Yeah, good. So this is the endoplasmic reticulum, so-called rough, because it has ribosomes, ATS ribosomes, stuck into it. They're not free. Okay? So we have this rough endoplasmic reticulum, rough ER. Good. And then this dude?
The mitochondria.
The
mitochondria.
Okay, right. Let's have a think about the functions then. Can we remember any of the functions? Plasma membrane?
Controls what enters and exits the cell.
Yeah, okay, so controls...
Oh yeah, of course you have to.
Controls entry and exit to cell, and you now have some idea of how that works. Okay, things like facilitated diffusion we talked about last term. Things like active transport. Okay, protein pumps that use ATP. These are all processes that are involved in controlling the entry and exit to the cell. Okay, what else? We've got endocytosis and exocytosis. All these things we learned about last term. So you should be thinking, yeah, along those lines, thinking, you know what it does now, but we've also studied how it does it. Okay? All right, so the nucleus,
Information.
yeah, it contains DNA. Okay, it contains DNA. All right, I'm not going to worry about this for now because I'll expect you to know what the nuclear pore does when we actually study it. So I'm going to leave that for now. Okay? And again, DNA we're going to get onto in grade 12, so we'll leave that for now. Cytoplasm.
Site of chemical reactions.
Good. So this is the site of chemical reactions. What does that actually mean? What sort of chemical reactions might take place? What should you be thinking about now?
Cellular respiration.
Good. So parts of cellular respiration. Glycolysis. Well done, Lewis. That's really good. The breakdown of glucose into pyruvate we studied last term. Yeah? Happens in the cytoplasm. What other things might happen in the cytoplasm? Think about what we started last lesson talking about.
Vesicle formation, maybe. Or...
So we've got exocytosis, endocytosis, movement of vesicles through the cytoplasm. But in terms of chemistry, chemical reactions, you're right, Lewis. You're correct. But I'm asking specifically about chemical reactions.
Hydrolysis.
Good. Hydrolysis and condensation reactions will take place here. Okay. So you should be thinking hydrolysis reactions and condensation reactions. Okay, every single chemical reaction is controlled by an enzyme. We're going to look at those next week. But every single biochemical reaction is controlled by enzymes. Each reaction has its own enzyme that controls it. Okay, so we could also add here that it contains enzymes. More on that next week. Okay, so mitochondria is the site of
Respiration.
one word I'm looking for, aerobic. Thank you, aerobic respiration. Well done. Okay, and that leaves us with the rough ER and the free ATS ribosomes. What do these guys do?
They're involved in proteins and everything.
Yeah, good. So this is the site of something called translation. Site of translation, which is a process we're going to study in grade 12. Okay, so the site of translation. Okay, and that is basically protein synthesis. So this is where proteins are made. Okay. So when you want to know where a hormone's made, where's adrenaline made? It's made in the adrenal glands, but specifically that hormone is manufactured on ribosomes. Immunoglobulins, rhodopsin, all these proteins that we've just looked at are manufactured on ribosomes. Okay? Either the free ribosomes or the rough endoplasmic reticulum. Now, what's the difference between those two?
The ones on the rough ER stay in the cell, don't they? And the ones the other way around.
Other way around, yeah. Other way around. Okay, but good. So the rough ER, this is where proteins for export. Who's doing economics? 3 out of 5 of you. Okay, cool. So export, use that kind of term. It literally is, it's a trade. The cell is making proteins that it gets its sells to the body, if you like. Okay, come out of the body. Okay, except they don't really sell them because your cells in your body kind of work as a kind of community that don't exchange money with each other in that way. Okay, they do specific jobs, but they're kept alive by other cells. If that makes sense. A bit like it's kind of the communist sort of I know. Anyway, yeah. So proteins for export. So this would be things like hormones. Okay, digestive enzymes. Sorry if you can't see this, I will move. Okay, they're all made. Oops, they're all made in the rough and then our free ATS ribosomes. This is where for proteins that are going to stay in the cell. Things like collagen that might be used to reinforce the cell structure. Okay, things like enzymes that are used in respiration, that are used in chemical reactions, will be made at the free ATS ribosomes. Okay, all right, and we will study that process in grade 12, both standard level and high level. We will look at, there's more to it at high level, but we will look at this actual process. How do ribosomes actually make proteins? And they do it by reading DNA. DNA contains the instructions to make proteins, which is pretty cool if you think about it. This is a molecule, right, that has information in it. Has information, tells your cell how to make other molecules, proteins. But more on that in grade 12. Okay. Any questions about that? Did anyone? So I'll leave this. I'm going to turn the board around. I'll leave this on. If you want to take a picture of it, okay. If you haven't had a chance to annotate your own work, did you get it? Did you get it right? Yeah. Yeah. See, Rhea's got some nice colour on there, which is good. It means you've been paying attention, listening. It's good. Okay. So, does anyone want to take a picture of that before I'm going to need the whiteboard again? But, no. You're going to have it on the live stream, aren't you? So you don't need to now, do you? Just put that on YouTube and you guys can just watch it again. Although I don't think you can see the rough ER down there, but you can hear me talking about it. Okay. So, we're going to look at then, we talked about last lesson, just before the break, that proteins have complex molecules, yes, called polypeptides, made from simple subunit molecules called amino acids. Okay? So when the ribosome manufactures a protein, what does it have to do you think? I'll give you a clue. Think about condensation again.
It must be a chemical reaction.
Good. So the ribosome is doing something to make a chemical reaction happen. What's it physically got to do?
Separate things, or
if you're going to build
things together. Yeah.
Okay. So we're going to build. So proteins are complexes built from subunits called amino acids. So if a ribosome is going to make a protein, it's got to join amino acids together. Okay? And it does that through our old friend, the condensation reaction again. Okay? All right. So, any questions about that? All right. You need to be able to draw this. So the next exercise that I've given for you here is to we're looking at understanding one. So amino acids are linked together by condensation to form a polypeptide. And we've got this first skill. It's an examined skill, okay? Skill 1, drawing molecular diagrams to show the formation of a peptide bond. So a peptide bond is the bond that joins two amino acids together. Okay? All right? Can anyone remember the general structure for an amino acid? Huh?
Like the straight one.
It is straight, but it's not the same as the fatty acid, which I think you might be thinking about.
Oh, it's a shorter one.
It's a shorter one, and it contains nitrogen. Okay, they all contain nitrogen. Okay, so I'm going to draw two amino acids. I'll show you how they form a peptide bond. Okay, and this is the basic reaction that takes place within a ribosome. Okay, so with our amino acids, I'm going to make sure I leave myself enough space here. We have nitrogen. Forms this NH2, this is an amine group, okay? Joined to a carbon atom, joined to another carbon. And that carbon has this structure. This is a carboxylic acid group. We have the same thing in the fatty acid. Remember the fatty acid tails, which are just carbon and hydrogen with this group at the end. The amino acid, carboxylic acid, fatty acid, amino acid. This is an acid group. Okay, COOH. Okay, so that's the acid, and then you have this amine group, the NH2 at the other end. Okay? Any questions? All right. The middle carbon has a hydrogen attached to it. Okay, now, individual amino acids, there's lots of different types naturally occurring. There's 80 to 100 naturally occurring, maybe even more, amino acids. They're all slightly different from each other. In living organisms, there's about 20 that are used in every single cell. Whether you're talking about bacteria or an oak tree or a human being, most life uses the same 20 amino acids. Another starting little piece of biochemical piece of evidence for evolution. Okay, when we get onto evidence for evolution, biochemistry is treasure trove. Okay, and one of the lines of evidence is the fact that this weird fact that all living things use DNA, DNA is universal. You can take DNA out of a bacteria and put it in a human cell can read it. It's universal. But also, biochemically, they use the same amino acids, about 20 of them. What makes them all different is what's joined to this central carbon. Okay? Now, we show that difference simply by putting an R there. Now, if you look at the periodic table, there's no element that is shown by an R. Okay? This just means radical. It's just this can be anything, basically. It can actually be anything. In the simplest amino acid, called glycine, it's just a hydrogen. Okay? But in more complex amino acids, you might get chains of carbon coming off here with maybe some sulfur and other elements put in there, iron and things like that. Okay? So we have this thing called an R group. Okay, so it's an R group. And that just means that it can be anything, and it's this part that defines an amino acid's properties. Okay? More on those in a minute. Any questions about that? Speak now, forever hold your peace. Okay. So what have we got to do to form a peptide bond? A peptide bond is a link between one amino acid to another. Okay? So we're going to add in another amino acid. So here's its amine group, H and H2, C, C, O, O, H. Okay, H and R. Condensation reaction involves the removal of what molecule?
H2O.
H2O. So we've got to somehow take an oxygen and two hydrogens off these two molecules to make a water molecule. Any suggestions?
You take the bottom ones off.
The bottom ones?
Yeah.
Good. Okay, so in this reaction, the oxygen hydroxide group here, the oxygen hydrogen, is removed from one amino acid, and sorry, I'll say that again. The oxygen hydrogen, the hydroxide group from the carboxylic acid end of one amino acid is removed, and then one hydrogen from the amine group of another amino acid is removed. Okay, so this comes off in this reaction to form H2O. And then what have we got left here? So we've still got an amine group at this end, HN. Okay, we've got, hello miss, we've got our middle carbon. We've then got our C double bonded to an oxygen there, but we've removed this. So this carbon, that bond is gone, that bond is gone. So this carbon is going to form a bond to that nitrogen. N there. H, C, C, O, O, H. Okay? So this is what we call our peptide bond. This is a covalent bond between or joining two amino acids. Simple? Or not? So you've got to be able to draw that. Okay? And you have space for it there.
So we're going to have to be able to draw this.
Yeah. You might be asked to. Probably be a three mark question. So wherever you see, a skill that says draw and label, that's something you're expected to be able to do. Yeah. This is the underlying principle of how your body manufactures the hormones that you need, the enzymes that you need, all of the kind of structural proteins as well. This is the basis of how your hair grows. And next week we'll talk about why dyeing your hair changes the colour, or bleaching rather, why that applying hydrogen peroxide to your hair actually changes the colour of it. Because that has to do with the proteins in the hair as well.
Isn't it the same thing of painting the nails?
No. If you were to apply, as I did when I had hair like that, red dye to it, then that's the same thing.
It was actually white before, but it came off.
And you need to use something called I think it's like purple rinse. Yeah, that's what old ladies use. Yeah. But it's so when you bleach it, what you're actually doing is you're actually changing you're damaging the protein basically, and changing the structure of the protein, so it changes. It goes white, and then if you were to add a dye to it, like blue or red or black or whatever, that would be like painting the nails. Yeah.
Wait, so people who have light hair, like blonde hair, it's easier for them to dye their hair.
Yeah.
They don't have to bleach it.
No.
So is there a difference between the proteins in their hair? And
No. It's just easier to apply red to a lighter background. If you or me tried to stick red dye into our hair, it wouldn't really show up.
Yeah,
It's
it's
too
your
dark.
reddish tint.
Yeah, exactly.
Exactly.
Why do people's hair colours change? Is it?
As they get older,
No.
like why do I go?
in general.
Why is Mr. Vincent going grey?
No, like in general, why do we all have? It's something to do with something else, or is it anything?
When you say change as you get older, or
No, like hair colour. How is it?
Sunlight as
Fine.
well has a big
Sunlight.
yeah, so
Or
the
salt
colour
water.
is to do with the protein that's in the hair and the light that it reflects.
Okay.
Okay, so the protein is absorbing some wavelengths of light and reflecting others. And if it's dark, it's basically absorbing lots and lots and reflecting maybe the kind of browny, wavelengths. The reason why people's hair colour is different then is that there's a slight difference in the structure of the protein that means it absorbs slightly different wavelengths of light and reflects others.
Okay.
Yeah, does that make sense?
That's cool.
Question. So when we're really, really little, we have different colored hair normally.
Does it tend to go darker, I think, as you get older, maybe?
I used to have like almost blonde hair.
Yeah. So yeah, again,
Yeah.
your proteins, it's just to do with the change in proteins. The same reason you go grey, I'm going grey, is the follicle cells in my hair on my head are losing their ability to make this pigment, this colour. That's a sign of aging, which is what happens. I started when I was 22. You know, what can you do about it? Any other questions? That's a good question. No? Okay. All right. So, if there's no more questions about that, the last thing I would like to do in today's class is to find out a little bit more about proteins. Okay, so there's a bit of a information sort of research exercise for you. We've got this exercise, facts about proteins. So this is addressing these understandings here. So that the fact that there's 20 different amino acids in polypeptides synthesized on ribosomes. Okay, so ribosomes make proteins, and proteins, there's 20 different amino acids that can be used to make up proteins. That's different to like starch. Starch is just made from glucose. Okay, but like hemoglobin will have different amino acids in it than insulin. Okay, because they have different functions, and that combination of amino acids changes their functions. You okay, Leo? Still with us? Yeah. I'm sending you to sleep. Talking too much on it. Don't worry, I'll give you a chance to get on with something right now. Okay, so this is, I've put in here, it's a link to topic 2.7 and 7.3. We haven't done them yet, and we won't do them until grade 12, but it's in there, so when you come back to review this, you can see that there's a link there. Okay? Amino acids can link together in any sequence. And we're going to do some maths to try and work out the probabilities of different combinations of amino acids. Okay, a protein may consist of a single polypeptide. Okay, so polypeptide, many, peptide. Peptide just means amino acid. Okay, polypeptide is lots of amino acids joined together. So a protein can be either a single polypeptide chain, or it can be lots and lots of polypeptides linked together. Okay, the amino acid sequence of polypeptides is coded for by genes. So the first one, I'm saying 2 and I'm saying 5, I'm not going to stress too much this week, because I know that when we get to grade 12 and we start looking at protein synthesis, we'll come back to this. You'll understand this when we look at the structure of DNA and how DNA makes proteins. So we'll come back to this in grade 12. Okay? Okay, so basically I've given you the answer to the first question. Hopefully you might remember. Okay. And I'd like you to work your way through these different questions. Now, this one I have told you, but if you want to use one of the textbooks to just kind of review it and write something in there, fine. Now, for this name 3 different amino acids, what I've helpfully put on my website is a table of, don't worry about what this codon amino acid assignments means. This bit, codons, we'll look at in grade 12. Don't worry about it. This is the genetic code, okay, and how it makes amino acids, but ignore all that for now. What I want you to be interested in is the amino acids. These are all the amino acids except for stop. That's not an amino acid, it just means stop. So from arginine up to isoleucine, these are all the amino acids. I'd like to pick 3 of those for the second question and find out what the R group for each of those 3 are and draw it into that space. Okay? I think the rest is fairly self-explanatory. Okay? And you've got the next 25 minutes to work through this exercise. Okay? Any questions? So just use this for question 2. Can you all see that? Yeah? Just pick. I've already told you what glycine is, for example. It's the only one I remember, annoyingly. It's like it's the only one I've ever needed to remember. Just it's the easiest one. And there's no, you're not going to be required to remember any more than the fact that there's an R group and what that R group means. You don't need to know the structure of any other, of any of these. You don't actually need to know that for your exams. What you do need to understand is that the R group can change. So if you get a multiple choice question, for example, that says, what is this molecule? And you see the amine group, NH2, C, and then there's a whole other load of stuff on there. I don't know, I've never seen anything like that. The clue is the fact it's got an NH2 group in it. Amine group means that it's an amino acid. You don't need to know which amino acid. Does that make sense? Just be aware that you could have all manner of weird and wonderful things instead of that R. Leo, does that make sense? Yeah? Okay, let's get to it.
Textbooks.
Textbooks in the usual place.
Okay. for me. So it's good.